Hello and welcome to another tutorial by Get Ahead Institute. So this video is going to cover part 2 of gametogenesis, which is oogenesis. So if you haven't subscribed to our YouTube channel, please do so and also leave a like. That helps the YouTube algorithm. Alright, so starting from where we stopped last time, just a brief recap. We said that gametogenesis is basically the formation of sex cells through the process of meiosis, reproductive cell division. Okay, so if the process occurs in males, it is called spermatogenesis. If it occurs in females, it is called oogenesis. So we covered the first part, which is spermatogenesis. You can check out that video uh, right here. But in this video, like we said, we're covering the second part, which is oogenesis. All right, so we are going to cover basically the embryonic development and the continuation of oocyte maturation and the stages together with is exploring what a mature Graafian follicle is. All right, so let's get right into it. So, all of the processes that are involved in transforming an oogonia to a mature oocyte is called oogenesis or ovogenesis. That's basically what it is by definition, textbook definition. All the processes involved in transforming oogonia to mature oocytes. So this process starts before birth and is completed after puberty at age 12 to 15 because these ladies are typically in the habit of maturing faster than males. Well, in males, spermatogenesis will start anywhere from 13 to 16 years uh, average. A sequence of events will continue only until menopause, and then after that, the process of oogenesis will cease, and that's why after that time, a lady is uh, no longer able to give birth or to form offspring, basically. So these events occur in the ovary and total completion of meiotic cell division that forms the female gamete is completed after fertilization. How does that happen? We will see as we get uh, further into this discussion. Okay, so oogenesis, embryonic development, where it all starts from. It all starts from right when the lady is still an embryo. So progenitor stem cells of a genetic female differentiate into oogonia. So why we mean genetic female is probably because of these days we have people who are becoming females, but we are not born females. So by genetic female, we're just saying people who are born with XX chromosomes. So the progenitor stem cells, which I explored in the previous video, the progenitor stem cells, these are cells that are going to commit to become uh, sex cells eventually. So in the female, the committed sex cells are going to differentiate into oogonia, and those oogonia will undergo a series of mitotic divisions so that they might increase in number a bit, and by the end of the third month, you're going to have them arranging in clusters, which these clusters are going to be surrounded by a layer of flat epithelial cells, which we'll be now calling as follicular cells. So when that happens, uh, we are going to call that a follicle, basically. So because they're surrounded by follicular cells, we call that a follicle. So asymmetric type of division occurs here as well, as mentioned in spermatogenesis, where some oogonia will begin meiosis but get arrested in uh, prophase 1 of meiosis 1. This is what is known as the first arrest. Here, the growth of these cells is stopped. Okay, and once it is stopped, like it's going to remain like that until puberty. That's when this arrest is going to be released. Okay, so these are before a lady is born, before a female is born. Let's call it a female. Before a female is born, the process would have begun. Okay, and it will be arrested somewhere and will be continued during puberty or when puberty starts. That's a difference, as in spermatogenesis, which all the processes will have to start at puberty. So there is that particular difference. So by month 5, the number of germ cells and oogonia reaches maximum. And at this point, you have all of the cells that the woman will ever need in her entire lifetime. They will be formed by this point. So, in other words, the lady has enough cells in her ovaries to last her entire fertile period all the way before menopause. She's not making any new cells by the time she's born. So, all of them are already born when she's are already there when she's born, rather. So, that's what we are trying to say here. So all of the surviving oocytes, uh, okay, before we get to that, pardon me. So there is a maximum number that is reached. It's a lot of cells, like millions and millions of cells, but not all of them are going to be needed, of course. Uh, some of them will have to, start to degenerate, and that degeneration forms what are known as atretic tissues. Okay, atretic tissues, they degenerate and become atretic in the ovary there. But then all of the surrounding oocytes that survive this cell death are in prophase 1. Like we said, the first arrest, they are in prophase 1 and they will remain like that, surrounded by the flat epithelial cells, and they will remain like that all the way up to puberty. That's when the other stages are now going to continue. So this arrangement, like we said, is called a follicle, yes, but what type of follicle exactly? It's a primordial follicle. It's a primordial follicle. All right, so that primordial follicle is what we are going to see in this picture expressed here. So we have a primordial follicle here. That means we have primary oocyte surrounded by the follicular cells there. The primary oocyte surrounded by follicular cells, flat epithelial follicular cells, and you call that a primordial follicle. So as this oocyte begins to develop further, you're going to see the epithelial cells turn a little bit more cuboidal, and they start to produce a certain secretion which will form a barrier known as the zona pellucida. Okay, so that's something that is happening here. So at the end of uh, month 5, okay, the first arrest has occurred. We are here at the primordial follicle. The growing follicle and the primary follicle, these come in during puberty. So that's what we are going to explore. All right, so now we have reached puberty, and now we want to continue. This lady was born, around 12 years ago, but then now she has reached ovulation period, which is 12 and beyond. So we have to continue this maturation of the oocytes. We have to unpause it from the prophase 1 of metaphase, the prophase 1 of meiosis 1. So the oocyte are arrested in the diplotting stage of prophase 1 until puberty. This arrested state is produced by oocyte maturation inhibitor OMI secreted by the follicular cells. So those follicular cells that are surrounding the oocyte here, they are producing OMI. This OMI is going to inhibit this cell, or maybe keep it in this paused state for all of those years. That's what is happening. So at birth, there is around 600,000 to 800,000 primary oocytes, and then degeneration will keep continuing, because at first, before the degeneration starts, we have a maximum, about millions, around 4 million, depending on which book you read. So that number keeps reducing until the time she's born. 600 to 800 primary oocytes. Degeneration continues. Only around 40,000 are present by the time she's born. So at birth, 40,000 oocytes are present at birth, because all of these just keep degenerating and keep dying. And 40,000 is still a lot, because a woman in her fertile period only needs about 480 if you do the math. From the time she's 12 and to the time she's, let's say, 45. Do the calculation, she needs around 400 to 480 eggs in that time. And the children that she's going to have are 5 or 6, depending on the type of person. So it's still in excess. The body still produces in excess, but all of that is just due to making sure that the chances are increased. Yeah. So the stages of oocyte development at puberty when you have resumed. So we were resuming from prophase 1 of meiosis 1. Don't forget that part. Prophase 1, meiosis 1. At puberty, that arrest is lifted and it continues. The production continues. So now when it continues, like we said, all of these are now happening after puberty, B and C. So cuboidal follicular cells are now forming. From these flat epithelial, they become more cuboidal, because they increase in number, become more cuboidal, and then start secreting the zona pellucida. This zona pellucida is going to be a barrier that is going to be important in certain functions that we'll explore further when we start doing uh, a fertilization. All right, so the primary follicle. So we are moving from a primordial follicle to a primary follicle. These two different are two different. Primordial before birth, before puberty. Then just after resuming, it starts growing into a primary follicle. The primary follicle now, you see that the zona pellucida is formed while these cuboidal cells have now become stratified. Okay, and there the zona pellucida is not only the only product that is produced here, you also have estrogens being produced by these follicular cells there. All right, that is what is happening at this point. So the stages of oocyte maturation after the primary follicle has been formed, there's going to be entrance of fluid into these spaces, the cellular spaces between the follicular cells. They are going to be fluid entrance, and then that fluid entrance is going to accumulate in a space known as a follicular antrum. Antrum simply means a space. So because the space is in between the follicle, it's called a follicular antrum. So this is going to be a fluid filled space in the uh, antrum, in the follicle, in the follicle. Yes. So it's still a primary follicle at this point, and it still maintains most of its uh, characteristics, except now at this point you're going to call the cells of the stroma, which are the you can call it the parenchyma of the ovary, who arrange themselves into what is known as a thicker interna and a thicker externa, although here they are exchanged. You have a thicker interna, which are the stromal cells that are closer to this follicle, and those that are on the outside, they are the thicker externa. These are the thicker cells that you are seeing there in yellow. These are just cells uh, that connect this structure to the connective tissue of the surrounding ovary, basically, or the parenchyma of the ovary. That's that. So we, like we have said, growing follicle, puberty, it grows into a primary follicle. The primary follicle gets fluid inside these follicular cells, and they that space is called the follicular antrum, and then that space will grow until it becomes something like this, and then there's a cell population that is found just above this uh, that is found just above this oocyte here. You're going to call that the cumulus ophorus. Very important structure to memorize in case you see it in MCQs. Very important. So that's just uh, the cells that are found there, of course. Okay, so these cells will later become known as the granulosa cells, or they are going to be called the corona radiator when it detaches from this uh, follicle at ovulation. Okay, so after puberty, the flat epithelial cells become more cuboid. This is just from what I was saying. Uh, yes, so this is basically what I'm from explaining. And then we have at the end having what is known as an antro or vesicular stage. Antro or vesicular stage is where this fluid field that I was talking about has developed. So the antro or vesicular stage is these two stages here. But then you have what is known as uh, a mature oocyte or the Graafian follicle. This is the Graafian follicle, a mature oocyte, which looks like that. So if you look at it, when more fluid accumulates in the follicular antrum, it's going to look like that. Even in a histological staining, actual picture of an actual thing, an actual cell, you're going to find that it looks like this. Here it's this the antrum is still accumulating fluid, and here it has accumulated the maximum amount of fluid basically. And then you see that particular arrangement. It grows, but then the antrum also grows with it. That means you have a lot of space here in the antrum, and that's how you know that this is a mature Graafian follicle in case they ask you what this is. Okay, so mature or Graafian follicles are basically what are present just before maturation, or rather, uh, are present at puberty, and they were the ones that sorry, let me repeat that. Mature Graafian follicles are the ones that are found just before ovulation. Just before ovulation, a surge of luteinizing hormone induces the pre-ovulatory growth phase. Meiosis 1 is completed. So, uh, the cell will enter meiosis 2, but it will be arrested again, which is the second arrest. Don't know why these cells have to undergo many arrests, but the main idea is to regulate the growth for whatever the reasons that the body does that, but uh, those growth phases are regulated through two arrests. So remember, don't forget the first arrest just before month 5, that is in prophase 1 of meiosis 1, and in metaphase 2 of meiosis 2. Metaphase 2 of meiosis 2, the second arrest happens approximately 3 hours before ovulation. Okay, so this word is supposed to be ovulation here. Okay, so two daughter cells are formed, but there is an equal sharing of the cytoplasm, and only one of them receives the most of the cytoplasm. The other daughter cell with little to no cytoplasm is called the first polar body. The polar body resides in the space between the zona pellucida and the oocyte membrane called the perivitelline space. So if you go back to this picture again, what we are simply saying is that after the antro or vesicular stage, the mature oocyte is going to begin meiosis 2, but it will be arrested at uh, the second stage, which is metaphase 2. Okay, so it will be arrested in metaphase 2. That's that growth will only continue if fertilization occurs. So if we remember properly from meiosis, we know that reductive division will be done after meiosis 2 is done. But at the end of meiosis 1, you have two daughter cells that are produced. At the end of meiosis 2, you have four daughter cells that are produced. So those two daughter cells is this same cell here, as it because it was being resumed in meiosis 1, because remember that it was being arrested in prophase 1 just after month 5, somewhere there. So as it is being resumed, you know that at the end of meiosis 1, you're expected to have two daughter cells. Those two daughter cells, now this is going to split, but the splitting is going to be very unequal, such that one of those daughter cells is going to get almost all the cytoplasm, and one of them is going to remain as a polar body, non-viable cell. Only one oocyte is supposed to survive at the end of the day. So that is going to occur, and then one of those is going to continue its maturation, and then it's going to be, as it starts meiosis 2, okay, as it starts meiosis 2, it's going to be paused again in the second arrest. And as we can see in these pictures here, let me just zoom in. So here we have primary oocyte in division, secondary oocyte, and polar body, and you have the polar body in division as well. So as you can see, this is where the arrest begins. Remember what happens in metaphase? In metaphase, the mitotic spindles are formed. So if you say in metaphase 2, that means this is what we are seeing here. The spindles are formed, the mitotic spindles are formed, both of both the secondary oocyte and the polar body will be in metaphase 2. Okay. Once that is paused, the only way this secondary oocyte can undergo or rather can finish this meiosis 2 is if fertilization occurs. If it doesn't occur, then this cell is lost just like that. The cell degenerates 24 hours just after ovulation if this is not done. Okay, so the place where it exists, this particular polar body, the other cell that is non-viable, that cannot produce, a child, so to say, is uh, which picture can I find? Okay, let's use this one for now. So there's a space known as the perivitelline space. You have the zona pellucida here, and then just before the zona, so there is a oocyte membrane, the zona pellucida, and then the follicular cells there. So in the space between the zona pellucida and the oocyte membrane here, there is a space known as the perivitelline space. This is where the polar body is going to reside when that cell divides. So when that cell divides, as you can see here, in the space between the oocyte membrane there and the zona pellucida is where the polar body resides. And there, at metaphase 2, that is now what is called the mature Oografian follicle. So we have at this point, we have talked about the primordial follicles found in just before birth. Then at puberty, it starts to grow into a primary follicle, which will develop into a secondary follicle, which has, the antrovesicular stage. And then after the antrovesicular stage, you will have now the mature Oografian follicle. So primordial, primary, secondary, and then mature Oografian follicles. This is what we have at the end of the day. Then when you have the mature Oografian follicle, that is basically the end of oogenesis. That means you have produced your oocyte. This oocyte, all it's waiting for is ovulation and fertilization for it to finish. Don't forget that it is arrested in metaphase 2 uh, before ovulation, and this can only be completed if fertilization occurs. So this is just a pictorial view of whatever is happening in the ovary from what we're from explaining. So a brief recap again. We have primordial follicles that you find just before puberty, and these are the only ones that you're going to find just before puberty. After puberty, you're going to be seeing primary follicles together with secondary follicles, and so these are primary follicles growing into secondary follicles as the fluid accumulates, and then at the end you're going to be having mature Oografian follicles, which are going to be releasing this mature oocyte at ovulation. So that's basically what is happening in this picture here. Primordial, primary follicles, secondary follicles. At secondary, this is where the antro stage or the vesicular stage fluid starts to accumulate in a space called the antrum, and then a mature follicle has a bigger antrum, and at the end of the day, it's called a mature Graafian follicle. Yes, other than that, that is the end of oogenesis, and we shall meet in the next video or the next session, which talks about the ovarian cycle leading to week 1 of embryonic development. See you.